In this video, we will discuss about the T cell activation by glycoconjugate vaccines, or in other terms, we will discuss how this glycoconjugate can be used as a part to trigger immune response within the host. And this glycoconjugate uh, vaccines would soon be one of the possibilities to treat several diseases. And so let us understand how this glycoconjugate vaccine works and activates the T cells. So let us first start with this polysaccharide that are present on its own, or polysaccharides that are present as a standalone entities. So it will generate only a temporary immune response in the form of IgM by this B cells, and it will not provide any informations to this T cells activation that can last for a longer time to target this kind of standalone polysaccharide standalone polysaccharides. So however, when this polysaccharide is combined with a protein, then it is known as glycoprotein. Now this glycoprotein will be detected by the BCR, or B cell receptors of the B cells, and this within these B cells, what will happen is that this endosome of this non-self glycoconjugate within the B cells occurs at the because of its interaction with B cell receptors, and after that the proteases, or digestion of this non-self glycoconjugates occurs within these B cell receptors, and thereby this major histocompatibility complex will take away some of the unique epitopes of this unique epitopes of these glycoconjugates, and then expose this and expressed this unique epitopes at the B cell receptors that are expressed at the surface with the help of this major histocompatibility complex 2, and these are known as self epitope. If there are some self epitope that are present in the glycoconjugates, then they will not be secreted on the expressed on the cell surface, whereas this self peptides that are found by major histocompatibility complex 2 will be degraded within the B cells. So only non-self epitope or non-self peptide will be expressed by the major histocompatibility complex at the B cell surface receptors, and from that these B cells the major histocompatibility complex at the B cell receptors will be interacted by this T cell receptors, or this is known as alpha beta T cell receptors. However, this alone interaction is not enough. They need other interactions also, so that that will provide the current scenario of this glycoconjugate within this host, and this information will be provided by this CD80 or CD86 interactions with this CD28 receptor, and this CD40 interactions with this CD40 ligand, and all these interactions in together will lead to the activation of the T cells in a way that it will generate antibodies against this non-self peptide that is expressed by this major histocompatibility complex 2 at the cell surface, and that will also lead to the secretion of this two things, that is interleukin 2 and interleukin 4, and this will act as a cascade effect in a way that this interleukin 2 receptors will interact with this interleukin 2 cytokines that are secreted by the T cells, and that will exaggerate the activation process of this T cells, and simultaneously this interleukin 4 released by the T cells will interact with the interleukin 4 receptors at the B cells, and that will also trigger the expression of the non-self epitope at the cell surface of the B cell receptors with the help of major histocompatibility complex. So both of these interactions and the release of the cytokines such as interleukin 2 and interleukin 4 will lead to the generation of the leads to the generation of the antibodies with the through these T cells that can target these glycoconjugates, non-self glycoconjugates that contains the non-self glycoepitopes or non-self epitopes. And simultaneously it will also make the memory T cells. It will also make the more memory T cells that will last within the host for a longer duration of time, so that in future whenever the host gets host get affected or attacked by this glycoconjugate vaccines, then the T cells then the memory T cells can directly act against this kind of glycoconjugates that have the non-self glycoepitopes. So this is how the T cells activations T cells can be activated by glycoconjugate type vaccines. If you need to understand more about this T cell activations using glycoconjugate vaccines, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.